Welcome to Biology A. We are going to be talking about special kind of proteins today. We call them enzymes as a large group, but there's a lot of different things that they do for us. So go ahead and get your guided notes out and ready to take notes for this lesson. It's very short. When we talk about enzymes, remember that we're still talking about the macromolecule group of proteins. So enzymes are still based on the building blocks called amino acids, but because they're so diverse and they're used in our body for so many very important processes, I'm covering them as a separate lesson. So just as a little bit of a warm-up, think back about proteins. We know that they're built on amino acids, like I said. There are like 20 different amino acid versions, so the amount of proteins that you can make from an endless combination of 20 different players is astounding. There are proteins that are the same across species, but there are also proteins that are different with specific species of different animals or organisms. The big idea remains, we're talking about using evidence to explain how carbon, hydrogen, and oxygen, and other specific molecules, or I should say elements, from the four major organic molecules we've talked about. So we covered carbs, lipids, and we're just wrapping up proteins today. We're going to finish nucleic acids later. So 3 learning targets that we're going to focus on in order for us to hit this goal is to explain how enzymes are proteins, which we've done. They speed up chemical reactions without being changed in the reaction. So think of them like reusable. So the primary purpose, once more time, for the first learning target, is that proteins speed up reactions and they're not used up. So they are reusable. Eventually they'll break down because, but describe how enzymes lower the activation energy of a chemical reaction. Now we haven't talked about this particular thing yet, but if you just look at the phrase activation energy, I think we can figure out what that means. Activation means to start, and energy, right, the amount of push it takes. So activation energy is the amount of energy it takes to get a reaction to start. And then finally explain how the environment may affect enzymes. So again, enzymes can be broken down. They're not used up in a reaction, but they can be broken down or broken based on things that are happening in their environment. So recap, making a protein starts with amino acids. We use small circles to represent these because it just helps us illustrate that they are like beads on a chain. If you put more than 2 amino acids together, that's a peptide chain, and each different color represents a different possible amino acid. There are 20. You can combine those in any way, and then, although we're not even going to talk about it for the test, a protein can fold upon itself once, twice, 3 times, up to 4 times to make these massive macromolecule complexes. But the sequence of which amino acids come next on this peptide chain determines the identity of the protein. So if I changed any one of these, it would be a completely different protein, even if there's thousands of amino acids in the complex. And again, this is not on your test, but if this sounds interesting and it sounds like something you'd like to learn more about, there's a video. I will post it for you. There's lots of things on the internet if you just wanted to like Google 4 levels of protein folding, for example, but again, not on your exam or your final. I just want you to know how complex proteins can get. And again, when you change the sequence of these amino acids, like for example, sickle cell anemia is a pretty serious condition. With sickle cell anemia, the normal shape of these red blood cells that we have floating around our body, when a person has sickle cell anemia, their red blood cells are shaped like sickles, or maybe bananas would be a more appropriate reference for you guys. But instead of having this nice hollow divot in the middle to hold oxygen, it's shaped differently, and there's no place for oxygen to bind. And what ends up happening because of their shape, they end up clotting or stopping blood flow, and that can cause some severe health issues. And if you'll notice, here's the normal chain of amino acids or peptide chain. And the only thing that's different in this one little spot is instead of a glutamic acid in sickle cell anemia, it's a valine. You're not supposed to have these memorized, so don't worry about that, but just understand that just one change. So just one change can affect its structure and then in the long run its function, because you know structure and function is a theme that keeps repeating ourselves in biology. The last time we talked about protein shapes, so keeping that in mind for structure and function. And just to reiterate, enzymes start or stop a reaction. So a chemical reaction would be you add two things together, and if you don't see something happen right away, sometimes you can add a special player called a catalyst or an inhibitor, and those would be enzymes, and then that would speed up your reaction. All of a sudden things are happening and things are really cool, but they wouldn't have happened without those enzymes there. And that's because enzymes have specific shapes to bind to specific other chemicals, and that brings those two things together when normally they might not be. They are not changed in the reaction, so you can reuse them, and they reduce the amount of energy needed to start the reaction. So those are the four key ideas about enzymes that you need to walk away with. So here's how this looks three-dimensionally for my artistic students. If this blue circle here is the enzyme, think of it like a lock, all right? The key being the substance it's going to bind to. It's very specific, just like any key in lock. Only something with this shape can fit into it, right? And then once that happens, it can accelerate the binding process that key would normally be going through. Here it is on a different schedule. Look, you can look at two different enzymes here. This one has two circular binding sites, and this one has a much more geometric binding site. So for this one on the left, these two if all four of these are floating around in the same solution or the same environment, these two molecules, the red and the green, they might not normally come upon each other unless they're bound into these specific spots. And once they're bound, if you brought the red one here and the green one here, they're right next to each other with their binding sites exposed, they're more likely to bind and make that happen. Likewise, the blue one goes over here and the purple one goes over there like Tetris, okay? It brings these other molecules, we call them substrates, into an arrangement where they can bind more easily. And that's how it speeds up chemical reactions. It can also stop a chemical reaction if, for example, it binds the two things permanently in the wrong way. So energy and chemical reactions, just like any energy in life, if you want something done, you have to pay something. Okay, nothing is free. So in the case of a chemical reaction, the reactants or the beginning ingredients, they are sitting at a certain energy. Even if it's burning something, even if this molecule here is a piece of wood and it's going to get burned, it's going to release energy. In order to make that happen, wood doesn't start on fire spontaneously. You have to give it something. You've got to give it a spark. You've got to give it some air. You got to whatever, right? So the process of making the reaction happens, there's a hump, there's a hill, and that's the activation energy. If you look at energy over the speed of the reaction. So you have to, this chemical reaction needs a certain amount of energy higher than it's already at. Could be heat, could be kinetic energy. Once these substances are given that, it's usually heat, then they'll be at a higher energy level, and that will engage the reaction to begin to go down, and the reaction will proceed. If you have an enzyme, oftentimes, look at the difference between this amount of energy required, so the difference between this dotted line and this one is much higher, so there's a greater activation energy without the enzyme. But if you have an enzyme involved, you'll have a smaller amount you have to pay, which will mean that the reaction will happen faster. If that's heat, for example, you don't have to heat it up as high. If it's a physical reaction, I should say kinetic energy, you don't have to stir it as hard. This is another example. It's just more of a cartoonish. So in this example down here, the enzyme and the substrate, without that, the dotted line would be the no catalyst, in other words, no enzyme. And then with the catalyst, the activation energy hill is much smaller. The enzyme and product are removed afterward. I just want to show you this to reiterate. It is not used up. It is free to go grab another substrate and start the process all over again. Enzymes are very sensitive, okay? They're sensitive to concentration. So they're only going to work as fast as you have something for it to work on. As soon as you run out of substrate, things are going to slow down. They're also sensitive to being what we call denatured. They can be physically, chemically broken down. So a high temperature or a high acidity or a high basic pH will change it. So for example, if you eat, sometimes you need to eat like specific probiotics and things because the normal reactions in your system are quite caustic. If you burn meat, for example, then that breaks down a lot of those proteins. Okay, and if you change the amino acid sequence, that changes the structure and the function. So there are 3 ways to change the productivity of an enzyme. That's to change the concentration of either the amount of enzyme or the amount of things to work on, substrate, to induce high temperature or extreme pH levels, or change the amino acid sequence. If you have too much change, that disrupts the bonds. So if this is the native or the original enzyme, if you, for example, denature it with heat or a high pH level, the shape of it changes. Now it doesn't have this active bonding site that we saw with the and that picture of the activation energy. You don't have that here anymore. You only have no site is a recognizable bonding site for the substrate. You do this all the time. If you have curly hair but you want straight hair, you will denature the proteins of the hair in order to get it straight. If you have straight hair and you want it curly, likewise, you just have to do it while you're wrapping it around something. So we do this all the time. So just check in with yourselves about how you feel about these learning targets. How do you feel about proteins as enzymes that speed up without being changed? Then describe how they lower the activation energy by bringing the 2 substrates together more often that wouldn't be able to be exposed to each other as quickly. And then they're affected by the environment by high temperature, extremes of pH, change in amino acid sequence, and if you have a different concentration. So if you have more or less substrate or enzyme. The more enzyme you have, the faster it will go. The more substrate, the thing it's working on, the faster it will go. And that's it. Thanks.